shows you that you're dealing with a lesion that is not growing as a normal tissue would. There's a lot of mitotic activity, so cells are dividing. You can see that by this mitotic figure here and this mitotic figure there. Again, in our body, cells are dividing all the time, but in most tissues you don't see them because they're still on a cell-per-cell basis quite rare. In tumors, these cells are dividing much faster. And another thing that is remarkable about this is that it looks like all of the cells that you see in here are of the same type of cell. They're all smooth muscle cells, but the liquid ones. In fact, what we have found is that per patient there can be different amounts of macrophages in these tumors, and that has a correlation in how well the patient will do and how aggressive the tumor is. And so we'll get to all that, but I just thought I would start with a little picture. And please also feel free to stop me if something's not clear. I've got an accent. I'm talking about stuff that you may not think about every day, and so just stop me if you want me to explain. So the goals of any, what they call translational research, that means research not so much to just only gather more information, but actually research with a goal in it to benefit patients, is, and this applies very much to leiomyosarcoma, is to find new diagnostic markers. So markers, proteins, that a pathologist can use to make sure that yes, this is a leiomyosarcoma versus no, it probably isn't. And that is something we're involved in, but we're more interested in finding LMS subtypes. And we all have the tendency to treat a disease once there is a name for it as if every patient is the same, as if every tumor is the same. And the more we know now about molecular studies, it shows you that many diseases actually have a number of subtypes, and that has best been shown in breast cancer from a study at Stanford that I was on a lower level involved in. And I actually want to do this in leiomyosarcoma. And why that is important, we will get to talk about in a minute. The second item there is to find new prognostic markers. So you would like to find features that you can determine on the cells, maybe a protein, maybe a type of messenger RNA, maybe a translocation in the chromosomes. It doesn't matter what it is, as long as it is something that correlates with the outcome of the patient, because that helps the clinician to determine the treatment for an individual patient. So for example, if there's a leiomyosarcoma that shows expression of a number of things that are associated with that outcome, then that patient would be more likely to be treated in an aggressive fashion by the oncologist than if those markers are lacking. Does that make sense? So, and prognostication, the process of making a prognosis, prognostication in leiomyosarcoma is very, very poorly developed, and we think we have found several markers that will help us prognosticate patients better. And then, of course, the ultimate and most important goal is to find novel therapeutic targets. Okay, so a lot of tumors these days are treated by sort of broad-spectrum chemotherapy, if you will. It's a drug that affects all growing cells, but because tumor cells grow faster, it affects them more. But it still affects the normal tissues, and that's why the side effects occur that you see in all patients. People lose hair, people become nauseous, and worse things can happen. People can get infections and whatnot. So what you would like to find is a drug that as specifically as possible targets to the tumor cells, okay, and just kills them and not the rest of the body. And that is the, depending on whether you're an optimist or a pessimist, it's either the pie in the sky project or something that is, hopefully doable in the next years. So, and I have to say, I don't know, I don't know where or diseases, waiting for somebody else to find a therapy on, say, breast cancer and colon cancer, and then hoping that it will apply to leiomyosarcoma is not a good strategy, because leiomyosarcoma is, together with many other sarcomas, its own unique disease with its own unique metabolic behavior, its own markers, its own prognostic markers, and it's unlikely that a targeted therapy, say, for breast cancer would work in leiomyosarcoma. In fact, the one targeted therapy that there is now in breast cancer, Herceptin against the HER2 protein, won't do anything with leiomyosarcoma because leiomyosarcoma doesn't express that protein. Is this about the speed that you can follow me, or?
Yeah.
Okay, just please stop me when I go too fast or am not clear. So, I would like to talk about 4 projects that we're doing in the lab. But before I do that, I just want to say some basic stuff about the techniques that we're using and also something about what cells are, what the cells are that make up our body. And so, first of all, a couple of slides about cells. So, a cell, this can be any cell, consists of a cytoplasmic membrane that keeps the whole cell together. The inside of the cell, which is the cytoplasm, where all the proteins are and all the organelles are. And then way inside, deeper inside there, what is called the nucleus. That's this thing here. And that's the location where the DNA is maintained. And the DNA, of course, is the genetic material that we inherit from both our parents. And that determines who we are. And cancer is, by most people, considered a genetic disease. So ultimately, there's damages in the DNA that get propagated into the rest of the cell that cause the cell to grow in an uninhibited fashion and become malignant. So the central dogma of biology is therefore that we have on the left-hand side the DNA, that is the data storage, if you will. It's all the data that you get, all the heritable data that you got from your parents. You can compare it, if you will, to a library of books. And the RNA is what comes out of that and what you look into and what you make active. And say, consider that, I'm just making an example, consider that you open a book on how to build a house. Right? Now you're learning about how to build a house, and then the protein part is where the RNA gets actually translated in protein, the actual building blocks that make the cell, or the house, if you will, the foundation, the siding, the framing. I'm just remodeling now, so I'm familiar with those terms. I wasn't before. So it really goes from DNA, data storage, to RNA, to protein. Ultimately, the proteins are determine how a cell behaves, what a cell does. If it's a cell in your eye, it will help you, transduce light images into something that you recognize with your brain. If it's a muscle cell, it will help you contract your muscle and move. Now, so we would like to know about all these proteins, but technically that is not really possible yet at the level of detail that is needed. So we are looking in our lab, we're looking at the RNA levels. And so we use that as a measurement for which proteins are abundant or not in a particular cell. Okay. And for that, and this started 10 years ago when Pat Brown invented these things at Stanford, we're using gene arrays. And I won't go through exactly how this works, but just let me say that at the end of the day, you end up with a big, or with actually a small glass slide, but under the microscope it looks big, with 40,000 dots on it, and each dot represents a gene, a human gene. And if that dot is turned red, that means that in the tumor there was a lot of RNA for that particular gene, and if it's green, it means that that particular gene had very low levels of RNA. And so what it leads to is that in a single experiment, these gene arrays can quantify the level of messenger RNA for over 23,000 genes in a single experiment. And that used to be, you used to have to do that on a gene-by-gene basis. So you used to have to do 23,000 experiments if you wanted to attain that level of knowledge. Now it can be done in one day. isolate the RNA, maybe to do cDNA, but the actual experiment is 1 day. So that has revolutionized really the amount of knowledge that people can get about any tumor, any disease. And that revolution is ongoing. And we are applying this to Lyme-Disease sarcoma. So what you can do with this is that you can not only say pick out one gene and say, okay, that's my gene, I'm going to work on it, but you can immediately look at which other genes are all going up together with it, or which genes go down when your gene goes up. And so you're getting a much broader view of what is going on in the cell. You're looking at patterns of gene expression. And this is a slide from about 10 years ago. You can see I didn't have a bald spot yet. And I'm basically holding down a paper here for Chuck Peru, who did a lot of gene array work on breast cancer. What we're holding out here, he, for the heck of it, once decided to print out all the information on 40 patients. And that's what it looked like. And this is fine print. So you can imagine that you're getting really vast amounts of data. Okay. So and actually with later technologies that we're now applying in the lab, later than gene arrays, the data sets become only larger. So that we're now looking at millions of data points. So one thing to do when you do research is that you find something and now you need to confirm it, right? You never go with one observation, nobody does, right? So you find something, you confirm it another time, now you start to believe it. Then you can start thinking about, for example, about could this be applied in humans, okay? We're not talking about one-day flights, we want to always confirm. And one way to confirm data from gene arrays is to use so-called tissue microarrays. And a tissue microarray is something that perhaps you are familiar with because a number of you, I believe, I don't know the names of course, but a number of you have contributed samples from Lyme-Disease sarcoma tumors that have been collected in our lab. And these samples come as individual paraffin blocks, like this one here or that one there. And what we do is we take a small core from a paraffin block and put it in a new paraffin block that is empty. Does that make sense? You just, with an apple core, but then small, like a millimeter, just punch a core of tumor out of the block and you put it in a new paraffin block. And you can do that 100 times or 200 times if you have 200 patients. And now in this one block, you'll have representations of 200 patients. You can say, well, big deal, who cares? Well, the thing is, it allows us to then look at many proteins with antibodies for many proteins in all 200 blocks in a very rapid and very cost-efficient fashion. If we would do an immunostable 200 block, that would be like, I don't know, maybe somewhere around $15,000 or something. If we do it on one slide only, it's about $15, right? So we can do many, many more samples much quicker with better controls and so on. So this is a very cool technology. It's not our technology, don't get me wrong, we didn't develop this. This is something that was developed by another group. But it's been extraordinarily helpful in our research, and this is how it will look under the microscope. So when a protein, in this case BCL2, is present in a particular tumor sample from, say, patient A, it will turn brown, and then this is patient B, who doesn't have that protein on the tumor, and it will not turn brown. And so you can therefore see patient A, high BCL2, patient B, low BCL2. You can compare that with your gene array data, and if it all matches up, then that confirms your